So I would like to start with a question. So, we're starting to talk about meiosis. And remember, one of our comparisons between mitosis and meiosis was that meiosis cuts the chromosome number in half, and mitosis maintains the chromosome number. So I'm going to pose a question to you. What would happen if sex cells, egg and sperm, went through mitosis and then fertilization, rather than going through meiosis and then fertilization? So I want you to ponder that question, and I'm going to open my chat. I want to start seeing some ideas pop up. So that's the question. What would happen if sex cells underwent mitosis and then fertilization? What would the result be if we went through mitosis rather than meiosis and then fertilization, the combining of an egg and a sperm? Okay, so I got a couple answers in here. Less cells would be produced because you're making 2 instead of 4. Wouldn't the genes be more random? Actually, if you're going through mitosis, which makes identical copies, they wouldn't be so random because they would be identical. What other ideas you got? How many chromosomes are in the human cell? 46. Very good. So 46 chromosomes are in the human cell. So when your sex cells go through meiosis, you cut the chromosome number in half, right? And we get 23. And an egg of 23 and a sperm of 23 come together and they make a zygote, a fertilized egg, of 46. 23 plus 23 is 46. If cells went through mitosis and you start with 46, how many would the daughter cell have? If you start with 46, you would have 23 pairs, so you'd have 46. So if an egg of 46 combined with a sperm of 46, how many does the resulting zygote have? Yeah, 92. So in order to keep the chromosome number the same through the generations, we have to cut the chromosome number in half before fertilization in order to maintain that 46, right? So that's the whole importance of meiosis, is being able to cut the chromosome number in half in order to maintain the chromosome number of the species. Okay, so today's topic, all about meiosis. Section 10.1 was asexual versus sexual reproduction, and then meiosis. And we're going to test all of this on Wednesday next week. So we need to get the details of meiosis going, right? So I would like you to go into the Google world. Go into the Google world to, you'll find Tuesday warm-up. And you should find a Plix, meiosis Plix. We've done these before. Last time we did it was glycolysis. It's a manipulative. 4 people have already done it. Okay, so we're going to do the manipulative and the 9 questions that follow. And remember that last question is usually a discussion question. So how could you discuss? Well, I could put you in a breakout room. So I'm going to create some breakout rooms for you guys to do the Plix together, but you each need to click through in order to turn in your own. So do the animation first and then the 9 questions. This is going to get us thinking about meiosis. A couple things in that video that animation you were looking at homologous chromosomes versus sister chromosomes or chromatids. So remember, the homologous are the ones that are similar. So I'm just going to jump right to this slide. Homologous chromosomes are similar chromosomes. They carry information for the same traits. So like maybe this one says blue eyes and blonde hair, and this one says red hair and brown eyes. So they're both carrying information for hair color and eye color. They're just not the same. The 2 purples, like the light purples, remember those are sisters. They're identical. So in that Plix, it was kind of zeroing in on when do you separate sisters and when do you separate homologs. So that's one of the things that we want to pick up in our lecture today. This produces variation. Mitosis does not, right? So your discussion question was, when would mitosis be a better reproductive option than meiosis? So I'm going to open up my chat box. If you could throw me some ideas here, or feel free to unmute yourself and just speak up. So I have one answer that it's like when organisms is bacteria or a plant. So like bacteria, single-celled organism, if it went through meiosis, it would cut its chromosome number in half. It wouldn't have enough genetic information, right? Plants, we know, reproduce through vegetative propagation often. And so again, if they cut their chromosome number in half, they wouldn't have all the genetic info they needed because they're not combining. They're not going through sexual reproduction. So they're not combining with another cell. So bacteria would benefit because it goes through asexual reproduction and it makes identical copies of itself. And so you want to keep those genes the same. Good. And I think you're getting into like the whole idea of the genetic information not changing, right? So the environment, if an animal is, or an organism is already well suited for their environment, why change their genes? It would be beneficial to them to go through mitosis because they're already well suited for their environment. So they keep their genes the same. Meiosis switches them up. So meiosis is good in an environment that's constantly changing, because variation means that some will be better suited for that area than others. Okay, so that was a quick little warm up to get us going. We have a lot of content to cover with meiosis. So we're going to get started on this. So now I'm on to, you have a lot of links today because of when I do them through websites, they launch individually. You want to open up the Zoom. So the Zoom has the PowerPoint I'm doing. It has a student version of the PowerPoint, and it also has a manipulative that Miss Hefty is going to walk us through at the end. Okay, so the student version, as before, it's there if you want it. So if you like to take notes and you want a way of making that go a little faster, you might open up that student version. If you like doing hand notes, don't open up that student version, no worries. Okay, so let's get going with meiosis, which prepares us for sexual reproduction. So meiosis is a form of cell division like mitosis. A few things are different. It halves the chromosome number, and it's only happening to produce reproductive cells like gametes and spores. So gamete might be a new word. It's one of those vocab words we covered Friday. Gamete is your egg or your sperm, depending on your gender. And then spores are produced in some other organisms like plants or fungus. So I'm going to skip my little links here because I know we're going to go long on time a little bit. So I'll put those links in later for you. A comparison here, remember mitosis is happening in somatic cells. Again, a vocabulary term. Somatic cells are body cells like skin, eye, hair, liver. They start with a diploid cell, 2 sets of chromosomes, one from mom, one from dad. They produce diploid daughter cells, diploid 2N, 2 sets. Creates 2 daughter cells, and they're all genetically identical. So we were just talking about that. Meiosis, today's topic, is used to produce sex cells, aka gametes, again, vocabulary word. Diploid and haploid, vocabulary. Diploid, 2 sets of chromosomes is what you start with. You will cut the chromosome number in half. So you'll only have 1 set instead of 2, and that 1 set then is called haploid. We refer to it as N. Rather than making 2 daughter cells, you make 4. You guys did a Venn diagram on your reading guide. So you probably identified these things. And you have unique combinations of chromosomes and some genetic recombination. Okay, so a lot more diversity. So this is what the picture looks like. So these are happening in what's called a germ cell. So like in the female, you have your ovary and you have your testicles, right? These are gonads. They produce gametes. So meiosis isn't happening in gametes. It produces gametes. It's happening in those gonad cells, and they're called germ cells. So when you see that word, that's what it's referring to. Meiosis happens over 2 divisions. So we're going to talk about meiosis 1 and meiosis 2. Please notice we're working with chromosomes that are paired sister chromatids in meiosis 1. In meiosis 2, notice we don't have the X's. So in meiosis 2, we're only working with sister chromatids. Okay? We have homologous chromosomes. The red and the blue carry the same information for the same traits. They're homologous. One is from mom and one is from dad. So homologous chromosomes in meiosis 1, sister chromatids in meiosis 2. So those are going to be keywords that help you like zone in on correct answers. They're both preceded with DNA replication. That's how you got the sisters, right? So you have interphase happening first. It doubles all the genetic info, so they're all now stuck with their copies. So a lot of this is going to sound like mitosis. It's just different groups of chromosomes going through these processes, right? So remember prophase 1, same as before. Spindle fibers formed, nuclear membrane disappeared, sister chromatids paired up. But now we get to talk about homologous chromosomes and tetrads. So this blue one, again, maybe carries blue eyes and blonde hair, and the green one maybe carries red hair and brown eyes. So information for the same traits, the information isn't identical. Maybe these two over here, notice they're different length. Maybe these two carry height and weight information. So these two are homologous with each other. They're both carrying height and weight. These two are homologous with each other. They're both carrying eye color and hair color. But these are not homologous to these, right? And I can tell because of their shape and size. So you have a synapsis, a coming together of a tetrad. A group of 4 is called a tetrad. Tetra is 4. So you have the 2 sister blues, the 2 sister greens. When they come together and form a foursome, that's called synapse. So synapsing occurs during prophase. That's a new word for us. It happens only in prophase 1. There is also some crossover that occurs. Notice the tangling of the green and blues. So pieces often break off during prophase 1, get reattached to other chromosomes, and that creates a genetic recombination. So notice this darker purple has a light purple arm. This lighter purple has a dark purple arm. So now, rather than saying blue eyes and blonde hair, it says blue eyes and red hair. And this one no longer says red hair and brown eyes. It says brown eyes and blonde hair. So you've recombined the genetic code. You need to know for the test when crossover occurs. It occurs during prophase 1. You need to know what it is, right? It's a genetic recombination. Pieces break off of one chromosome and reattach to its homologue. Metaphase, remember they were lining up at the equator. Same thing's happening, just we're talking about a tetrad now rather than a single chromosome. So pairs of homologous chromosomes line up at the equator. They are attached at their spindle fibers. Remember, the spindle fibers move the chromosomes throughout the process. Again, I'll link that later. So this is just reiterating the homologues. Notice the size and shape. These are different than these, right? So when we look at a karyotype, when we look at a micrograph of all your chromosomes, we know which ones pair up because they're the same length. They have the same centromere position. They have the same banding pattern that is basically their genetic code, okay? So homologues are similar, not identical. Sisters are identical. Anaphase 1, those pairs are separating, okay? So you have homologous chromosomes separating, sister chromatids stay together. So anytime we talk about homologues, we're talking about meiosis 1. So homologues separate and move to opposite poles. Again, the spindle fibers are doing the work. At this point, you no longer have 2 sets of chromosomes, right? At this point, you're dividing and you only have one set of chromosomes. So we went from a diploid cell to a haploid cell. That is a test question. When does reduction division occur? During anaphase 1, when the homologs are separated. So telophase, again, the cell is going to pinch in the middle with cytokinesis. We're going to form two now haploid daughter cells. My cat wants my salmon spread. Okay. I gotta hide it, sorry guys. Okay, so we've now finished meiosis 1, right? We have two haploid daughter cells. We no longer have homologs, we only have sisters. So throughout meiosis 2, we talk about sisters. This is very much like mitosis, right? So single chromosomes are visible. A new spindle fiber will form, connect with all of those centromeres. Those spindles will align the chromosomes at the equator. And then anaphase 2, the sisters will separate. Okay, rather than homologs, the sisters separate. Centromeres, remember that's the point that the two sisters were connected, so those divide, allowing the sisters to be pulled to opposite poles. Telophase, we now have 4 cells because you had 2 and each of those split in half, so now you have 4. Notice they all have half the original chromosome number. You started with 4, you now have 2, a long and a short. Notice they're not one of these are the same. You didn't get a long green and a long blue. You didn't get a long, I'm sorry, and a short green. You got recombination happening, all kinds of different cells. So the nuclear membrane reforms around each of these, and the cytoplasm divides. Meiosis. You've created 4 haploid gametes. Okay, and I'm going to skip the practice for a second. This happens differently for males and females. So I'm going to show you just a quick something about each of those. Okay, so spermatogenesis, hopefully you can guess that makes sperm, right? Male gametes. And oogenesis, ovum are eggs. So the oo is referring to the eggs. So the genesis, formation of, so this is the formation of eggs. So this is only happening in animals. I'm going to skip that slide for now. Spermatogenesis is going to create 4 sperm. They are all haploid and they will all survive. Spermatogenesis does not begin until puberty. This is meiosis. It's just a specific version of meiosis. So not till puberty will male animals begin to make sperm, and then they'll make several hundred million sperm every single day, which is just a great, like you don't make that many white blood cells a day. So it's just crazy to me. And this is how it would work. So here's your 2N original cell, right? You divide, here's your 1N because the first division we reduction. And then the second division, we still have 1N. So those are your 4 daughter cells, even though they're male, they'll gain a tail later. Females, we're going to find are different because not all 4 of these will survive. So in oogenesis, you start with that primary oocyte, and this happens before the female is ever born. So remember, the males didn't start this till puberty. Girls already are in meiosis before you're born. All the eggs will stop. They're stalled out at prophase 1. Then puberty, meiosis will begin again. It'll take you through the first division. So you finish that meiosis 1 and you make 2 oocytes. One of them will die off, and I'll show you a picture of this. So we call the ones that die off a polar buddy. I believe that was one of the vocabulary terms. So only one survives. That secondary oocyte goes through the division again, and once again, one of those will die and one of those will survive. This stalls out again at metaphase 2. So at metaphase 2, that cell will be like that goes through then ovulation. the whole menstrual cycle thing. So that cell is kicked out of the ovary, and then it'll only finish meiosis if it's fertilized. So females may not, like the majority of your eggs never go through all of meiosis, whereas the males are going through all of meiosis, each one of them. So what they're doing, start here again, right? Here's your 2N. Notice this cell is very little and this cell is much bigger. So this cell doesn't have enough cytoplasm to survive. So cytoplasm has a lot of nutrition and information in it. So the secondary oocyte kind of hogs all that cytoplasm because this is like life's goal. Nature's goal is survival of a species. So this gives the greatest chance to support a developing fetus. So only one of these two cells will go through meiosis 2. Again, it divides unequally. One is tiny and one is big. So again, the ovum hogs the majority of the cytoplasm and the polar body will die off. So in oogenesis, only one daughter cell survives. So that's a big difference between males and females. Here you can see the male version, all 4 surviving. Here you get a better picture of those polar bodies. And then this is only the one that survives. So males and females, just a little bit different. Overall, 2 important things you want to remember about meiosis is that it cuts the chromosome number in half. That makes it so that after fertilization, chromosome number doesn't change. Also, it rearranges the genetic code. So every daughter cell is different. So it makes genetically unique daughter cells. You already created a Venn diagram, so you don't necessarily need me to go through this again. Both are preceded by DNA replication. So interphase happens first. Mitosis has 1 division, meiosis 2. There is no synapsing in mitosis. There is synapsis of homologs in meiosis. You make 2 daughter cells for mitosis, 4 for meiosis. For us, for animals, we use mitosis for growth and repair and replacement. Meiosis we're using to prepare us for sexual reproduction. Okay, you should be able to compare the 2 processes, even though we did mitosis in semester 1. You should be able to identify phases of meiosis based on images. Okay, and at that point, I'll be turning this over to Miss Hefty because we're going to work with those images. Okay.
All right, so these are the 4 options for meiosis 1. Which one did you guys decide was the first step? And you can just type in the chat like A, B, C, or D in order. So like this is A, this is B, this is C, and this is D. Getting a lot of B messages, so we'll call that one first. And then what one did you guys put for second? And then how about third? All right, so it looks like the orders you guys came up with was this, which is perfect. You guys rock. This one has way more options, so I think I'm just gonna walk you through this one. So we're gonna have 2 different things going through meiosis 2, right? Because we ended this with 2 cells. So we'll start again with 2 cells. And then they're in prophase 2. And then they move on to metaphase where they all the chromosomes line up in the middle, right? And think of this like this is one arrow going down this line and this is one arrow going down this line. So it's happening twice. And then they start to pull apart from the center and the cells begin to separate and you end up with 4 gametes. Is that how you guys did it? You can type in the chat. All right.